Just that, right? Good morning, everyone. In the room, in the room and online. I want to say thank you for the invitation to this conference and also for the support to early career scientists on their journey to independence through these awards. They're a great support for us. And a research postdoctoral, a postdoctoral researcher in St. George's University of London in the group of Professor Ostergaard. And today I'm going to present pretty much is a work in progress project on mechanistic studies of EPHB4 pathogenesis for lymphatic anomaly treatments. In St. George's, we are a multidisciplinary research team that have established along the time a bench-to-bedside approach with the focus on primary lymphatic anomalies. And historically, the group has been led by Professor Ostergaard, and our interest has been looking for the genetic causes of lymphatic anomalies. In the team, we have two bioinformaticians that are Sarah and Lilin, that are in charge of the exome and genome sequencing, variant filtering, and finding those variants. And in 2014, I was joining the group to build up a functional genomic team that they will take forward this work to perform the functional validation and then also mechanistic studies. And now Cassin and Ruby are joining me in this team. Our work wouldn't be possible without our clinic. Is led now by Dr. Christiana Gordon, and we are a national referral clinic with clinicians, nurses, and especially therapists. And we work very close also with another referral center in the NHS and the University of Derby. And part of our work also involved the development of new imaging techniques, lymphoscintigraphy, ICG, and MRL, to help us define better the phenotypes and also to get insights into the pathophysiology of the disease. And this diagram, I think that reflects very well our goals towards improved diagnosis and new treatments, but combining our expertises in phenotyping, genotyping, and mechanisms. And in this sense, as you heard already about, what are my interest topics are developing and implementing in vitro models for patient variant modeling and functional validation, and these molecular mechanisms to find new treatments through drug repurposing. Key to being able to find genetic causes for primary lymphatic anomalies has been for us the development of a classification algorithm that we follow in St. George's that help us to group the patients within the clinic based on the phenotype and the possible genotype cause. And today I'm going to bring your attention to a group that is characterized by patients that have a systemic and visceral involvement as presenting with generalized lymphatic dysplasia, and specifically EPHB4 associated lymphatic-related hydrops. That is the topic of my talk today. And we become interested in EPHB4 it's a receptor tyrosine kinase because we found there were mutations causing autosomal dominant related lymphatic-related hydrocephalus. We found two families, one in the UK and another one based in Norway. And there was the main clinical characteristic of these families. They were presenting with hydrops fetalis, that is defined as excessive fluid that is present in at least two fetal compartments. And those two families have a long history of intrautero and neonatal deaths, as you can see in the pedigrees. They were presenting some members with some cardiac defects and also some venous problems. Only one of the patients that we see in our clinic in the UK was clinically presenting with lower limb lymphedema, but when we performed lymphoscintigraphy in this patient and other family members, and from both families actually, we saw that there was a common feature of a problem with absorption of the tracer and also rerouting. So there was definitely a lymphatic problem in these families. And since then, there's been other clinical phenotypes that are being associated with pathogenic EPHB4 variants, as we heard before, capillary malformation or arteriovenous malformation syndrome 2, or central conducting lymphatic anomaly. And also in the last years, we've been also contacted by many clinical diagnosis centers looking for an answer about novel EPHB4 variants that are of uncertain significance. So they wouldn't know whether they were pathogenic or not. So we got into the task of looking about whether those variants were pathogenic, and then also we expanded also the clinical phenotype associated with EPHB4, finding 5 additional cases of fetal hydrops, and one was presenting with pleural effusion, so we see in the scans here, and also edema in the head. And also we found 2 families that were presenting with telangiectasias and capillary malformations that we had never seen before in our clinic in the EPHB4 cohort. We enroll in the task of try to address those comments about are those variants causative of the disease pathogenic. So we overexpress then on lymphatic endothelial cells that we stimulate with EphB4, that is the ligand that activates the receptor. We perform immunoprecipitation using a tag, and then look at the phosphorylation of the receptor as a readout of its activity. We compare all our variants with the wild type healthy protein, and then we use also 3 SNPs. So there are single nucleotide polymorphisms that we found in the general population to be on a high allele frequency. So we were thinking that there could be controls for benign variants, and in fact we found that in these cases when we stimulate the ligand with the ligand the receptor, we could see activity with this increase of the signal as we were finding in the wild type, but most of our variants was lacking this activity of the receptor. Apart from one of them that we couldn't confirm then in our assays whether it was pathogenic or not. So based on our model, for the majority of our the variants that we see in our cohort, we know that the receptor must be present in the membrane of the cells, but the mutation affects the activity of the phosphorylation of the receptor, and then it's going to have an impact in the forward signaling. And we don't know yet whether there could be a haploinsufficiency of a dominant negative effect. And we have here today or yesterday a lot about the interaction of the link of EPHB4 with the RAS-MEK pathway and the promising possibility of the use of MEK inhibitors on this pathway. So we are also asking whether we could potentially use MEK inhibitors to treat our patients that carry EPHB4 variants. So the specific aims of the project that was awarded was the generation of CRISPR-Cas immortalized lymphatic endothelial cells that will carry EPHB4 variants as a new in vitro tool in my lab for modeling patient-derived mutations. The first step is the characterization of these immortalized lymphatic endothelial cells. I will present some preliminary data, including the optimization of a live imaging technique. And that will be followed by the development of CRISPR-Cas protocols. And the second aim is this cellular signaling pathways that could be dysregulated as a consequence of these EPHB4 variants, with the further objective of identifying possible drugs that could be tested in vitro to correct lymphatic function. We started to perform a mass spec experiment in lymphatic endothelial cells, looking for novel interactors. And once the CRISPR cells are generated, I would like to perform a high throughput screening for MEK inhibitors using in-cell Western technology. A couple of years ago, Frenkel and colleagues developed an immortalized lymphatic cell line and a 3D microfluid model to use for functional assays. So they use, the company is based in the Netherlands, it's called Mimetas, and it uses a chip, a plate that contains 40 chips, when we can grow the immortalized lymphatic endothelial cells in the top channel, close to the extracellular matrix, in this case collagen, and we can apply some growing media to these chips. And after a few days in culture, a tubular structure develops. Here is the staining with bicadirin, and by adding the specific lymphogenic factors to the channel, the cells can sprout, and those sprouting can be also imaged and quantified. And another useful item of this is that can be used to perform barrier integrity assays. So in this case, they wanted to test the integrity of their tubes. So they played the immortalized lymphatic endothelial cells or a control that has no cells on. And after a few days, if you add physit dextran in the top of the channels, you can see that when there are no cells, that there's a leakage of the physit, and that could be also quantified and measured. So the first step for us is do a characterization of the lymphatic identity of the cells, and we are collecting samples at early and late passages and comparing also with primary legs that we use in the lab, and we will analyze different markers by immunofluorescence and Western blot. So we start testing that those cells actually express before, and then also other lymphatic markers. And this case is a test that we did when we were growing those cells at passage 11. So keep in mind that we use primary lymphatic endothelial cells up to 5 or 6 passages. So that already elongates the ability of grow those cells in culture. They were able to grow those cells, they say, up to 52 passages, that is 12 months. We want to characterize also the response of VGFC, comparing also these two cell types and early and late passages. And we are doing that by RNA sequencing, that we have the data back now and doing the analysis at the moment. And we want to develop also this assays, this live site imaging, that we've also a high throughput method to analyze VGFC-driven proliferation and migration. So I think that is all could be very useful, as I say, to compare cellular characteristics between two cell populations. And we are starting also to try and test primary lymphatic endothelial cells versus immortalized cells, plus and minus VGFC conditions, and we can follow, in this case, proliferation is measured as the increase in the total dry mass along time, and also we can track the speed and the displacement of the different populations. We would like also to use that in the future to compare the impact of the variants on cell proliferation once we have these CRISPR-edited cells. But we started to see whether there were differences when we use a system that overexpressed our EPHB4 wild type or one of our variants using an adenovirus delivery system. And we can see in this condition, we're comparing the wild type in a medium that contains different growth factors, but no VGFC. And then when we add VGFC in red, you see an increase in the proliferation of the cells along time. But in the case that we overexpress one of the EPHB4 variants in the patients, we don't see this difference, like they don't not responding to the VGFC. But we saw what we saw actually, because when we compare the two, the wild type and the variant in the absence of VGFC, is that the variant, it seems to have a hyper-proliferative phenotype, because you can see increase in the number of cells compared with the wild type along time. In terms of the relationship between EPHB4 and the ERK signaling pathway, our preliminary data has been performing a human phosphoarray to look at the impact of activation of the pathway on 43 kinases at the same time. We confirm a decrease in the phosphorylation of ERK, as others have already shown, and this is also corroborating the standard Western blot. But then we saw that when we overexpress one of our variants, that is, this is one of the first families that we published, the Norwegian family that I presented at the beginning. It seems to be a hyperactivation of ERK, and then matches with the hypothesis of suggesting MEK inhibitors as potential therapeutics. Finally, we have also started to look for novel interactions of EPHB4 in lymphatic endothelial cells, and we perform mass spectrometry. We treat the cells with control or the ligand, plus or minus VGFC also, to look for interactions between the two pathways. And this is just a matter of putting together some numbers reflecting the changes that we see related to increased binding or decreased binding detected, comparing the different situations. But we now need to do proper bioinformatic analysis of that. So as I say, as you can see also, is a work in progress, and the future work will involve conclude this immortalized cell characterization. Hopefully we can share the findings with the community, and follow by the development of the protocols that I hope to start in the new year. Related to the mass spectrometry project, we are now starting the bioinformatic analysis to find what will be the candidates to take forward to experimental validation. Then we will assess what is the role of these novel interactors with EPHB4 in lymphangiogenics, and of course see what happens when we overexpress our variants instead of the wild type protein. As I say, once we have the CRISPR-edited cells, I will perform a high throughput screening for MEK inhibitors using cell Western blot to see what is the impact in the signaling. And this has been already put forward as a part of a grant application to award in the UK to be able to get more funding to progress this project. I will hear about that in February 2024. And the last bit of my plans is bring and optimize this 3D lymphatic nanotube technology in the lab to perform the functional mechanistic studies. And it's also part of our application for a PhD fellowship that we have put to the British Heart Foundation, and that will be hopefully hear positive news next month about that. I want to say thank you to St. George's team, especially to Kasim, Sarah, Ruby, and Lillian are the people that are involved in this project, to the participants and families, of course, and funders, past funders that help us to start our work in EPHB4, and my current funders that are helping us to progress this research. Thank you so much.